Today we are going to discuss about animal cell culture. So this is the learning objective of today's session. After this session you will be able to understand what is the definition, what are the different types, and what are the different steps of animal cell culture and what are their applications of animal cell culture. So let's start with the definition of animal cell culture. So we had already discussed about different culture methods in bacteria where we had used some kind of culture media to grow some bacteria, right? So that is a method for culturing of the bacteria. But in order to culture an animal cell, it is much complicated than an culturing of a bacterial cell. So it required some culture media and it required some controlled artificial environment. So we can define animal cell culture or simply cell culture as a process by which cells are grown under the controlled condition, generally outside their natural environment. That means in an controlled artificial environment. Because usually we know animal cells will be grown on the different organs or different tissues of the animal, right? Suppose if you are taking examples of skeletal muscles, that will be grown on the skeletal muscle itself, skeletal cells. Now, if you are taking myocardial cell, that will be grown on heart. So these are the natural environment. But in case of animal cell culture, we are going to culture these cells of animals in an outside of its natural environment. So in order to do this, uh, each cell have different condition. So the condition for each cell will vary, but generally they need some suitable vessels in which we are going to grow the given cells. We need some media that will supply essential nutrients like growth factors, hormones, gases like carbon dioxide, oxygen, and also we have to control some physiochemical characteristics like pH, osmotic pressure and temperature. So these are the requirement of the animal cell culture. Now what are the different cells which is used for animal cell culture? So first thing is we have to remove the specified cells from the animal in order to grow in an outside environment, and which also requires some favorable condition. And this removal can be either by directly, as in case of surgery, so by using some case of incision or by using some surgical procedure, we can separate the cells, or we can use some kind of mechanical methods like sonicator or ultrasound or vibration. So these are the different methods for separation of the cells from the organs, or by using enzymatic action by using some enzymes like lysozyme or trypsin. So these are the different methods to separate or to take the cells from the organs of the experimental animal. So we cannot take the cells from all the animals. We have to take these kind of cells from some specific experimental animals. So this can be obtained by previously made cell lines or cell strains. So there are some procedure to preserve the already obtained previously made cell lines. We can preserve that as a cryopreservative method. Okay. Now these are some examples of cells which can be used for animal cell culture. Please don't think we can culture all the cells, like we can culture the uh, skin, we can culture the different kinds of liver cells or etc. There are some specific cell that only can be cultured as an animal cell culture. So these are some of the example. First one is fibroblast, second one is lymphocytes, then cells from cardiac as well as skeletal tissues, then cell from liver, breast, skin and kidney, and also we can culture different kinds of tumor cells. So tumor cells is an important cell which is used in the animal cell culture because we can we can multiplicate it in short period with uncontrollable manner, and we can subculture, we can culture at any because these are immortal cell, right? Cancer cells or tumor cells are immortal cell. So these have a wide application in case of animal cell culture. Tumor cell have a wide application in animal cell culture. Now, what are the requirements for an animal cell culture? So first requirement is media. So while discussing on the media, please understand the media which is used for the bacteria is not enough for culturing the animal cells. It should containing some more complex media than the minimal media which is sufficient for the growth of microorganisms. Now we can classify the culture media which is used for animal cell culture into natural media and artificial media. So first one is natural media. So as the name indicate, this media is isolated from natural sources. So there are different natural sources like blood clots, coagulant or biological fluid or tissue extract. Sometimes we can use this plasma clot or coagulant, which is usually available as liquid plasma in silicone ampule or lyophilized plasma. So as we know, there are some procedure for blood transfusion. So that are usually collected in the silicone or what kind of rubber or some kind of plastic covers, right? So after the expiry of these kind of blood products, we can isolate their liquid plasma from the expired blood of which is taken for the transfusion. സാധാരണഗതിയിൽ ഏറ്റവും നല്ലൊരു സോഴ്സ് എന്ന് പറയുന്നത് ബ്ലഡ് തന്നെയാണ്. ആ ബ്ലഡ് സാധാരണഗതിയിൽ എടുക്കുന്നത് ഇതുപോലെ ട്രാൻസ്ഫ്യൂഷന് വേണ്ടിയിട്ട് നമ്മൾ ബോട്ടിലാക്കി വെച്ചിരിക്കുന്ന ബ്ലഡിന് അതിൽ നിന്ന് പ്ലാസ്മ സെപ്പറേറ്റ് ചെയ്തെടുക്കുക എന്നുള്ള പ്രൊസീജിയറുകൾ ചെയ്തിട്ടൊക്കെയാണ് നമ്മൾ എടുക്കുന്നത്. ഓക്കെ. Now there are some other sources like biological fluids, which is isolated from in the form of sera from human blood, or from the plasma cord blood or horse blood, or in the form of biological fluid like amniotic fluid, ascistic fluid, or coconut water, or insect hemophilus serum, aqueous humor from the eye, or culture filtrate. So these are the different uh, sources for natural media for the animal cell culture. Now coming to the artificial media again, in artificial media, along with the natural source, we have to add some kind of organic or inorganic nutrients, vitamins, salt, serum protein, carbohydrate, oxygen, carbon dioxide, gases phase. So these are the different uh, artificially added materials. Okay, so that will not be there in the natural media which containing blood and other serum. So we have to add some kinds of hormones like cortisone, testosterone, or insulin. We have to add some kind of growth factors like platelet derived growth factor and fibroblast growth factors. And we have to add some kind of proteins like fibronectin, albumin, and transferrin. So all these are the different sources of artificial media. So we have to add some kinds of hormones, growth factor, or protein into the serum containing media. Sometimes we may have to use this material in a serum free media. So serum have some problem with their specificity. So in order to make an selectively for particular cell type, we may have to use some kind of serum free media. So we have to take this kind of hormone growth factors and protein in another serum free culture media. Okay, so now what are the different complex media or artificial media? So here along with the serum, we may have to add salt as well as glucose or amino acid as well as vitamins. So we have to add 9 essential amino acid with different amino acid names: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. In addition to that, we may have to add some other amino acid, non-essential amino acid like cysteine, glutamine, and tyrosine. Okay, so these are the different amino acid which is required for an media used for animal cell culture. And so this is about the media. Now in case of growth factors, we may have to add some kind of severe critical growth factor which is required for critical regulator of cell growth as well as differentiation. For example, for the important functions of skin fibroblast, we in order to proliferate when needed to repair the damage resulting from the cut and wound. So this deficient is usually triggered by the growth factor released from the platelet during the blood clotting. So we had mentioned one growth factor, that is platelet desired growth factor, which is very essential for the growth of this kind of platelet. Okay, so the identification of individual growth factor has been made possible. Uh, the culture of the variety of cell in the serum-free media, and usually the media in which serum has been replaced by some specific growth factor required for the proliferation of cell in question. So whatever the specific cell we are going to take for animal cell culture, depending on that, we may have to change the serum free media or sometimes serum containing media, etc. Now these are some of the optimal conditions for animal cell culture. Along with the culture media, we need to give some artificial controls. Usually, mainly the culture of the animal cell is depend on the temperature. So the deciding of the temperature is purely based on the animal from which the cell is taken. ഏത് തരം അനിമൽസ് നിന്നാണ് ഈ പറയുന്ന സെല്ല് നമ്മൾ എടുത്തിരിക്കുന്നത് എന്നുള്ളതിനനുസരിച്ചിട്ടാണ് ഇൻക്യുബേഷൻ ടെമ്പറേച്ചർ നമ്മൾ തീരുമാനിക്കുന്നത്. So if the cell is taken from an warm blooded animal, we have to use 37 degrees Celsius for the incubation. So if we are using some kind of cold blooded animals, we have to take 20 to 25 degrees Celsius as an incubation temperature. And we have to keep the osmolality of between 260 to 320 milliosmolar per kg, and we may have to use some kind of buffer to stabilize the pH, and also we have to add some exogenous carbon dioxide, and we have to increase the viscosity by using carboxymethyl cellulose as well as polyvinyl pyrrolidine. So these are the some environmental condition which is required for the culture of animal cell culture. So this is all about an introduction and different media used for the animal cell culture. From this, the media required for animal cell culture is an important question which is usually seen in your university question paper. So please note that media required for the animal cell culture and as well as examples of animal cells which is used for the animal cell culture is very very important from this topic. Now coming to the different types of animal cell culture, we can classify the cell culture based on the number of division into two, that is primary cell culture and secondary cell culture. Again, this primary cell culture is again divided into adherent cells, suspension cells, and confluent cell. And again, the secondary cell culture is again divided into finite cells as well as continuous cell lines. So let's see one by one. So first one is primary cell culture. So as the name indicates, they are the first cells. They are the first cells obtained from straight from the cells of host tissue. So once we are isolating the cell either by surgical procedure or by mechanical procedure or by enzymatic methods, we will separate some cells from the animals or host tissue, and these cells are cultured in an uh, optimum media with optimum condition. So these cells dissociated from the parental tissue, which is grown in a suitable container and culture, is known as primary cell culture. Usually they are heterogeneous. Different cells will be different, and most of the cells have an limited time of life, limited lifetime. Okay, it will die within a few minutes or few hours, and almost most of the cells are very very similar to their parent cell also. So this is the property of an primary cell culture. Now based on the primary cell culture, when they are grown in an vessel, based on the growth of these micro or cells, we can divide again into adherent cell, suspension cell, and confluent cell. So first one is adherent cell, as the name indicate. These are the some cells which is adherent to the vessel which is used for the animal cell culture. So these are also known as anchorage dependent or propagate, and they are propagating as a monolayer. ഇതൊരു single, if you are taking a flat vessel for an animal cell culture, these cells will adhere to the surface of the vessel and they will propagate as a monolayer. അതിന്റെ മുകളിലോട്ടൊരു ലെയർ വരില്ല, ഒരു സിംഗിൾ ലെയർ ഓഫ് സെൽ ആയിട്ടായിരിക്കും അത് വരുന്നുണ്ടാവും. So need to attach to a solid or semi-solid substrate for their proliferation. So that is a property of adherent cell, and these adhere to the culture vessel with the use of extracellular matrices, which is generally derived from the tissue of organ that is immobile and embedded in the network of connective tissue. So example for an adherent cells is fibroblast and epithelial cell. So these cells are usually can cultured only as an adherent cell culture method. Okay. Now, if when the bottom of the culture is covered with a continuous layer of one thickness, they are known as monolayer. So culture വന്ന് അതിന്റെ ആ വെസലിന്റെ ബോട്ടം കംപ്ലീറ്റ് ആയിട്ട് സെല്ല് കൊണ്ട് കവർ ചെയ്ത് നിൽക്കുന്ന രീതിയിൽ, ഒരു ഗ്യാപ്പ് ഇല്ലാത്ത രീതിയിൽ നിൽക്കുന്ന സമയത്ത് ആ ഒരു കൾച്ചറിന് പറയുന്ന പേരാണ് monolayer culture എന്ന് പറയുന്നത്. And the advantage of this adherent cell culture is this can be directly transferred to an slip or slide to examine under the microscope. So this is very very important to understand the histopathology or histology of each cell during cell culture. So we have to analyze the changes which occur in the animal cell culture. So this kind of cell is very very easy to transfer into a slide and to examine under the microscope. That is adherent cell. They are anchorage dependent. They will adhere to the surface of the vessel which is used for the animal cell culture. Now coming to the suspension cell, as the name indicates, they will be suspended in the culture media which is used for the animal cell culture. So they are anchorage independent. They are non-adherent. They will float in the culture media. They do not attach to the surface of the culture vessel. And examples include hematopoietic stem cells, which is derived from blood, spleen, and bone marrow, and tumor cell are example for suspension kind of cell. Example for adherent cell are fibroblast and epithelial cell. And example for suspension cells are hematopoietic cells and tumor cells. So hematopoietic cell, which include blood cells, spleen, as well as bone marrow. And the advantage of this kind of cells are they will grow much faster and they do not require any kind of replacement of the culture media. So that is the one of the important advantage when we are going to perform an animal cell culture. We need a short period of time to culture the cells of suspension kind of primary cells, and usually they are homogeneous type. An enzyme treatment is not required to dissociate from that, and they culture have a short lag period. അതിന് കുറച്ച് സമയം എടുത്തിട്ട് മാത്രമാണ് അതിന്റെ നമ്പർ കൂടുള്ളൂ. Okay, so that is a suspension cell. Now next one is confluent cells. These are the cells which will get when an suspension cell overgrown. So what will happen if there is any growth, there is more growth in case of this kind of suspension cell, that will be overcrowded, and they will occupy all the available substrate or substrate. They will reach confluent. There will be some kind of mixture or some kind of aggregates of cells. So it will be very difficult to differentiate that. And if we didn't keep or if we didn't subculture this kind confluent culture, the growth will be detrimental. So growth will be reduced. എന്താ സംഭവിക്കാന്ന് വെച്ച് കഴിഞ്ഞാൽ ഇങ്ങനെയുള്ള സമയത്ത് ഗ്രോത്ത് സസ്പെൻഷൻ സെല്ലിനെക്കാളും കൂടുതൽ ഗ്രോത്ത് വരുന്ന സമയത്ത് they will not have any available substrate to occupy their space, so they will start to aggregate. Okay, അത് കട്ട പിടിക്കാൻ തുടങ്ങും, ഓവർ ക്രൗഡഡ് ആവാൻ തുടങ്ങും for their container, so the further growth will be detrimental. So it is very essential to subculture a portion of the cell which is transferred to a new cell which provide more space for the nutrient and for continual growth. So the cells which is taken from this kind of cells to get another set of cells is known as subculturing. That is secondary cell culture. So these confluent cells are cells which is isolated from the organ itself. Then these are the first culture. Okay, ഒരിക്കൽ ഓർഗന് നമ്മൾ ഐസൊലേറ്റ് ചെയ്ത് കൾച്ചർ മീഡിയയിൽ അതിന്റെ ഒപ്റ്റിമം കണ്ടീഷൻസ് ഒക്കെ കൊടുത്ത് കൾച്ചർ ചെയ്യുന്ന സമയത്ത് നമുക്ക് കിട്ടുന്ന 3 ടൈപ്പ് കൾച്ചറുകളാണ് ഇവിടെ പറഞ്ഞത്. അഡ്ഹറൻറ്റ് സെൽസുകൾ, അത് വെസലിന്റെ സർഫസുമായിട്ട് അഡ്ഹർ ചെയ്യും. സസ്പെൻഷൻ സെൽസുകൾ ആ വെസലിൽ സസ്പെൻഡ് ചെയ്യും, ഫ്ലോ ചെയ്ത് നിൽക്കും. കൺഫ്ലുവൻസ് സെൽസുകൾ അത് അഗ്രിഗേറ്റ് ചെയ്ത് നിൽക്കാൻ തുടങ്ങും. അഗ്രിഗേറ്റ് ചെയ്യുന്ന സമയത്ത് ഗ്രോത്ത് കുറയും. ഈ സമയത്ത് ബാക്കിയുള്ള ഫേസിൽ നമുക്ക് സബ് കൾച്ചറിങ് മാൻഡേറ്ററി അല്ല, നെസസറി അല്ല, ബിക്കോസ് they can further grow up to this confluent growth. So after this confluent growth, the growth will be decreased. So it is very essential to subculture after the confluent growth of animal cells. Okay, so that is primary cell culture. Now what is secondary cell culture? So once if you are subculturing from the primary culture, they are known as secondary culture or secondary cell line or secondary subclans. Okay, so നമുക്കൊരു പ്രൈമറി സബ് കൾച്ചർ കിട്ടിയിട്ടുണ്ട്. ആ പ്രൈമറി സെൽ ലൈൻസ് എന്ന് നമ്മളൊരു സെല്ല് എടുത്ത് വീണ്ടും അതിനെ സബ് കൾച്ചർ ചെയ്യുമ്പോൾ നമുക്ക് കിട്ടുന്ന സാധനത്തിനെ പറയുന്ന പേരാണ് സെക്കൻഡറി കൾച്ചർ ഓർ സെക്കൻഡറി സെൽ ലൈൻ കൾച്ചർ എന്ന് പറയുന്നത്. This process involves removing the growth media and dissociating the adher cell and subculturing of the primary cells to different divisions lead to generation of cell lines. So this will be different from the original parent cell. Secondary cell will be different from the original parent cell because it will show different kinds of property based on the physical environment we are providing. If you are changing the growth media, if you are changing the physiochemical parameters, they will act different from the original cells. Again, this secondary cell is again divided into finite cell as well as infinite cell or continuous cells. So this is based on the lifespan of the culture. Once if you are doing subculturing of secondary cell culture, on the basis of the lifespan of the culture, we will classify the secondary cell culture into finite cells and infinite cells. So in case of finite cell, the cell lines go through a limited number of division with a limited lifespan. So that kinds of cells are known as finite cells. So after several passage of time, after several culturing, they will lose their ability to proliferate. So this is a genetically determined event of losing of its ability to proliferate. That is known as senescence. Okay, so that is known as senescence. So the finite cells have a limited lifespan. After the limited number of cell division, they will lose their ability to proliferate and they will dead. So this is a genetically determined event that is known as senescence. Now what is mean by continuous cell lines? So suppose if we are subculturing the secondary culture media or secondary culture, it will undergo some kind of transformation and acquire some kind of ability to divide independently or indefinitely. So that kind of cells is known as continuous cells. So what will happen during this kind of continuous cells? They will become immortal. So after an 70 to 80 subculturing, they will get an property or they will get an ability to divide indefinitely. They will become immortal. മരണമില്ലാത്ത സെല്ലുകളായിട്ട് മാറുക എന്നുള്ളതാണ്. അപ്പം അങ്ങനെയുള്ള സെല്ലിനെ പറയുന്ന പേരാണ് കണ്ടിന്യൂസ് സെൽ കൾച്ചർ എന്ന് പറയുന്നത്. These cells are less adherent, fast growing, less fastidious in nutritional requirement and able to grow up to a higher cell density and different in phenotypes, and they will grow more in suspension. So this have a more advantage, more application in animal cell culture, this kind of continuous cell culture. Okay, so this is the different types of animal cell culture. So we had classified the animal cell culture into primary cell culture and secondary cell culture, again based on the property of they are adhering or not. The primary cell culture is again divided into adherent cell, suspension cell and confluent cell, and again secondary cell, which are the cells isolated after subculturing from the primary cell culture, again divided into finite cell culture. They have limited lifespan, and they another one continuous cell lines, they are immortal cell, they will divide indefinitely. Okay, now coming to the different steps of animal cell culture, so we can categorize the steps of animal cell culture into three. First one is isolation of the tissue, second one is disaggregation of the tissue, then third one is seeding of the culture into the culture vessel. So first one is isolation of the tissue. So already we had mentioned we will use the cells of experimental animals like mice, rabbit, guinea pig, etc. And then we can use mouse embryo or other kinds of uh, things for the animal cell culture. We can use some kind of mouse embryo for undifferentiated fibroblastic culture. And also we can use muscle cell or alveolar cell or macrophage, leukocyte cells. All these cell can be used as an primary or cell for animal cell culture, and it has to be stabilized with 70% alcohol, and it has to be removed aseptically without contamination of any kind of microorganism. Now coming to the disaggregation of the tissue. So it is not possible to isolate a single cell from an organ in first step itself. So here we are going to isolate the tissue, right? We are going to take a myocardial tissue or even mouse embryo, muscle tissue or alveolar tissue. So from this tissue, we have to disaggregate the cells. So tissues are composed of cells. So these cell has to be disaggregated, and this can be done by different methods like physical disaggregation by sieving, syringing, or vigorous pipetting, or by enzymatic disaggregation, or by using chelating agents. For enzymatic disaggregation, we can use some enzyme like trypsin or collagenase. So trypsins are of two types, cold trypsin as well as warm trypsins. Now coming to the second step, that is the disaggregation of the steps. So this can be done by some kinds of mechanical method or by using some kind of enzyme, and it has to be washed with a physiological buffer, and it has to be subcultured if required for secondary cell culture. Now the third step is seeding of such culture into an culture vessels. So the dissociated primer cell grown in the primary cell culture is seeded on the culture plate at an high density for secondary culture. So we have to use different culture media for this kind of animal cell culture. We have already mentioned different requirements of medias like amino acid, vitamins, glucose, bicarbonate buffers, growth factors, hormones has to be used. And in order to avoid the contamination of the microorganism, we have to use some kind of antibiotics like penicillin, streptomycin. It must be added to prevent the contamination of the microorganism. And in case of mammalian cell, which is an cold-blooded animal, so sorry, for warm-blooded animal, so we have to incubate at 37 degrees Celsius. So if you are using any kind of cells from cold-blooded animals, we have to keep the temperature of 15 to 26 degrees Celsius. So this is the different requirement for animal cell culture, and always we have to maintain the aseptic techniques. We are going to perform an animal cell culture, so we are going to culture the specific cells of animal, but along with this culture, the bacterial infection should not be there. So bacteria like mycoplasma, fungal infection are the commonly possible contamination in the animal cell culture. So it must be done in a sterile environment. It must be done in HEPA filter or laminar airflow unit with a constant unidirectional flow of HEPA filter in order to avoid the contamination. And also, if required, we can preserve the cell which is cultured as an cryopreservation. So hope you have aware about this cryopreservation method. We have to use some kind of cryoprotective agents like dimethyl sulfoxide or glycerol, and we have to store at minus 130 degrees Celsius in liquid nitrogen. So that is the cryopreservation method. So we will store the cells which is cultured from animal for further use in cryopreservation. Okay, so this is preservation of the animal cell culture method. So this is the procedure or steps of animal cell culture. First, we have to isolate the organ after getting the ethical committee clearance from the ethical committee for sacrificing the experimental animal. Then we have to take the specific cell fragment or tissue. Then by enzymatic digestion of the cell fragment with the enzyme trypsin of collagens, we will get the single cells, and then we will culture the single cell in a suitable culture media, that is known as primary cell culture. Now we can culture this up to 70 to 8 times for and secondary culture. So this time it is known as established cell culture. So in syllabus there is a term like primary cell culture and established cell culture. Okay. So the difference between primary cell culture and established cell culture is, once the cells which is isolated from the organ is cultured in a suitable culture media, that is known as primary cell culture. So once if you are taking a primary cell culture to subculture up to 70 to 80 times. So this type of primary cell culture is known as established cell culture. Why? Because up to this round of times, 70 to 8 times of subculturing, this primary cell culture have a limited lifespan. They will be die after a specific time. So that is why it's known as established cell culture. Now what will happen after the 70 or 80 times of subculturing? They will alter their cells, cell division characteristics. They will alter their chromosomal number, and they will acquire some property of indefinite growth property, immortal property. That kind of cell is known as transformed cell culture. These cells are transformed from the established secondary culture or primary cell culture to an immortal cell or indefinite cells. So that's why it's known as transformed cell culture. So this is one of the questions usually seen in the question paper. What is the difference between primary cell culture, established cell culture, and transformed cell culture? So in case of transformed cell culture, they are the cells which is isolated after 80 time of subculturing, and these cells have a property. They have some alteration in their cell division characteristic. They have some alteration in the chromosomal number, and these cells will acquire a property of indefinite growth property. That means they will get an immortal property or indefinite cell division property. That kind of cell is known as transformed cell culture. Now, what is established cell culture? These are the cell culture which we will get after the primary cell culture up to 70 to 80 subculture. അപ്പോൾ ഇതൊരു സ്ഥിരമായിട്ട് ചോദിച്ചു കണ്ടിട്ടുള്ള ക്വസ്റ്റ്യൻ ആണ്. എന്താണ് പ്രൈമറി സബ് കൾച്ചർ, സെൽ കൾച്ചർ, സെക്കൻഡറി സെൽ കൾച്ചർ, എസ്റ്റാബ്ലിഷ്ഡ് സെൽ കൾച്ചർ അല്ലെങ്കിൽ ട്രാൻസ്ഫോംഡ് സെൽ കൾച്ചർ എന്ന് പറയും. അപ്പോൾ പ്രൈമറി സെൽ കൾച്ചർ എന്ന് പറയുന്നത് ഏതാണോ ഓർഗൻ, ആ ഓർഗനിൽ നമ്മൾ സെല്ല് ഐസൊലേറ്റ് ചെയ്തിട്ട് ആദ്യമായിട്ട് കൾച്ചർ മീഡിയയിൽ അനിമൽ സെൽ കൾച്ചർ ചെയ്തപ്പോൾ കിട്ടുന്ന സെൽസിനെയാണ് പ്രൈമറി സെൽ കൾച്ചർ എന്ന് പറയുന്നത്. ഈ പ്രൈമറി സെൽ കൾച്ചർ നമ്മൾ സബ് കൾച്ചർ ചെയ്യുന്ന സമയത്ത് അപ് ടു 80 ടൈംസ് ഓഫ് സബ് കൾച്ചർ ചെയ്യുന്ന സമയത്ത് കിട്ടുന്ന സെല്ലിനെയാണ് നമ്മൾ എസ്റ്റാബ്ലിഷ്ഡ് സെൽ കൾച്ചർ എന്ന് പറയുന്നത്. ഇനി 80 ടൈംസിന് ശേഷം നമ്മൾ സബ് കൾച്ചർ ചെയ്യുമ്പോൾ നമുക്ക് കിട്ടുന്ന സെല്ലിനെ പറയുന്ന പേരാണ് ട്രാൻസ്ഫോംഡ് സെൽ കൾച്ചർ എന്ന് പറയുന്നത്. ഓക്കെ, so this is all about the steps of animal cell culture. So we will take the tissue, then we will take the animal cell, specific cell, then we will culture in a specific nutrient media. Then we will add to an primary cell culture. Then if required, we will subculture it and we will get a secondary cell culture. Okay, so that's all about the steps of animal cell culture. And finally, the application of cell culture. So this is very, very important. This is also a repeatedly asked question for university examination. So there are different applications like vaccine production. So we had discussed about the reproduction of a virus or cultivation of virus. So there are three methods. Since the viruses are obligative parasite, we cannot use culture media to culture the virus. We have to use either an animal cell or we have to use an embryonated chicken or duck here. And third one was animal cell culture. So animal cell culture is the important source of multiplication of the viruses. So in order to produce some viral vaccines like polio, rabies, chickenpox virus, hepatitis vaccine, measles, we require to culture the viruses. So in order to culture the viruses, we will use this animal cell culture to continuous growth of this animal to produce the virus production and also for the production of the vaccines from viruses. Second one is cancerous research. So again, cancer is one of the important disease which we are getting affected by most of the population, or especially some of the elder age. So the cell lines, instead of studying the property of a cancer in an human subject, it is better to take the animal tissue to external environment and to test what are the different causes of cancer, what are the different drugs which can be used for the treatment of cancerous cell, and how they are acting on the cancerous cell. So why this normal cell as well as cancer cell also can be grown in the culture media. So we can study the basic difference between this normal cell as well as a cancer cell if we are performing the animal cell culture. And we can also study different uses of chemicals, viruses and radiation which can be induced to produce or which will causes the cancer, and we can study the different mechanism of that causing the changes in the cancer cells. And we can also determine the suitable drugs and method of selective destroying of the cancer cell, other than we are going to study some kind of property in human subject itself. Okay, so it will be very useful for studying cancer research. And next one is virology. Again, once again, virus can be cultured in the animal cell culture. So this method can be used to that kind of property. Now, next one is cell-based manufacturing. Again, there are lots of pharmaceutical products which are synthesized from the cell-based protein engineering or genetical engineering, like antibiotic, monoclonal antibiotic, insulins, hormones. All these are produced by biotechnological process. So these kind of products can be used by genetic engineering by using animal cell culture. So we can use synthesis or production of the replacement of different tissue as well as organ. Artificial skin can be used for treating burns and ulcers, and artificial organs also under the study like pancreas, liver, kidney. All these are under the study of cell lines. And also this can be used for the production of viruses by using virus production like polio, rabies, chickenpox, etc. Now, next one is genetic counseling. This is one of the important area. Much researches are going on where one doctor can identify different genetically produced fetal disorder by taking the sample from the uh, fetal cells. So, aminosynthesis is a diagnostic technique that will enable a doctor to remove and culture the fetal cell from a pregnant woman, and it is an important tool for early diagnosis of fetal disorder. There are so many genetic disorder which can be identified during this fetal stage itself. So, പ്രസവിക്കുന്നതിന് മുന്നേ തന്നെ ജനിക്കാൻ പോകുന്ന കുട്ടികൾക്ക് എന്തൊക്കെ രോഗങ്ങൾ ഉണ്ടാവുമെന്നൊക്കെ സാധാരണഗതിയിൽ predict ചെയ്യുന്നത് ഈ cell culture method യൂസ് ചെയ്തിട്ടാണ്. The cell can be examined for the abnormality in their chromosome, their gene using karyotypic, chromosome painting and other molecular techniques. Now, in genetic engineering, again, genetic engineering is the alteration of genes by using genetic engineering or biotechnology. So here the ability of transfect or reprogram culture cell with a new genetic material has been provided major role to molecular biologists. So can be used to produce new proteins in large quantity. So insect cell, which is used as a miniature of cell factory to express the essential quantity of the proteins. Again, genetic engineering have more application in the uh, animal cell culture. So since we don't have to sacrifice any animals or any organs or anything, we just need some kind of cell lines so that we can study this kind of genetic engineering procedure. In gene therapy, here the ability of genetic engineering cell also led to use of gene therapy. So there are some diseases which is caused by genes. So by modifying the gene, we can avoid or we can prevent the that kind of occurrence of the diseases. So the cell can be removed the patient lacking of some functional gene, and missing or damaged gene can be replaced by this kind of cell line studies. The cell can be grown for while in a culture and then replaced into the patient. So this is what we will do in the gene therapy. We will isolate the gene, the lacking gene in the human, and we will culture that gene in an animal cell culture, and we will replace that lacking gene into the human, so that we can prevent different kinds of diseases which is caused by genes or genetic disorder. And drug screening as well as development, once again, it is very important to study different effect of the drug before using in an animal or human subjects. So the experiment in human as well as animal subject is very difficult, and it is very complicated procedure to get approval for this kind of procedure. So before starting the animal as well as human study, it is very mandatory to perform cell based assays, not just for the toxicity testing but also for thought of screening of the compound. So if we suppose we are going to discover some kind of new drugs, and we don't know the pharmacological effect of that kind of drugs, so we have to do some kind of preclinical study as well as clinical study. Preclinical study in the sense in animals, and in clinical study in the sense in human. So before starting itself, we can screen the effect of the drug in the cell lines without sacrificing any animal or human. Okay, so that's all about the animal cell culture. We had discussed about the definition, different culture media used for the animal cell culture, different types of animal cell culture, procedure for steps of animal cell culture, and application of cell culture. So hope it is clear. So that's all about the animal cell culture.